Okay, let's continue with chapter 3, molecules of life. So this will be 106 life molecules 3. Okay, we ended, I think, talking on the last one about proteins that are polymers of amino acids. Now we're going to go to the last macromolecule, or large molecules of living things, nucleic acids. Macromolecules that store information and provide the instructions for building proteins. So they're informational macromolecules. And they are polymers also, polymers of things called nucleotides, which we're going to see in a moment. And there's two types, DNA and RNA. DNA stands for deoxyribonucleic acid, and RNA stands for ribonucleic acid. And they have different structures and different functions. Basically, it's DNA holds the information, and RNA is used to access the information. And the information is the blueprint, the instructions for making a protein. That's what a gene is. Okay. Genetic material that humans and all their organisms inherit from their parents consists of giant molecules of DNA. And the DNA resides in a cell as one or more very long fibers called chromosomes. And eukaryotic chromosomes are wrapped in histone proteins. And that substance is collectively called chromatin. And it's found in the nucleus. So a gene, unit of inheritance, encoded in a specific stretch of DNA that programs the amino acid sequence of a polypeptide. Amino acid sequence, what amino acid goes in what order to make a particular protein. Right? Those program instructions are written in a chemical code that must be translated from the nucleic acid language to the protein language. So this is called gene expression or protein synthesis, making a protein from the instructions in a gene. And it consists of two parts, transcribing the gene, making an RNA copy of a DNA gene, and then translating it at the ribosome into an amino acid sequence. So there's two languages, nucleic acid language and protein language. That's called translation, because it's going from one language to another. So there's the information superhighway there. DNA holds the information in genes. It's opened up, and the RNA copy is made of one of the strands. This is called transcription, because transcribing is just copying something, right? That RNA is taken out of the nucleus, brought to a ribosome, and a protein is constructed from the information in a coded form on the RNA. So, nucleic acids are polymers, as I mentioned, from monomers called nucleotides. Each nucleotide, these monomers are a little more complicated than the other ones. They have three parts. A 5-carbon sugar. In DNA, it's deoxyribose. In RNA, it's ribose. A phosphate group, and a nitrogen-containing base, or a nitrogenous base. And there is a nucleotide. There's the sugar, in this case it's deoxyribose. The phosphate group is attached to one of the carbon on the left, and the nitrogenous base is attached to the sugar on the right. Now, nitrogenous base could be one of 4 different bases: adenine, guanine, cysteine, and thymine. And they all begin with this different letters. So usually you often just use the letter, and we can put it in a symbol form, having the sugar, phosphate, and then the base. In this case, it's thymine. And the information in DNA is not found in the sugar phosphate backbone. It's found in a sequence of nitrogenous bases. That's where the information is. And it's still Okay, so each DNA nucleotide has one of 4 possible bases: adenine, guanine, thymine, or cytosine. And there they are. And there's a space-filling model of DNA. Notice that it's a double helix. It's a long, thin molecule that's a double helix. There are the nucleotides again, or dehydration reactions. So again, you're going to use dehydration to link nucleotide monomers into long chains called polynucleotides. Form covalent bonds between a sugar of one nucleotide and a phosphate of the next. That's why it's called a sugar-phosphate backbone. Sugar, phosphate, sugar, phosphate. And then the bases hang off the backbone like appendages. And you can think of it as a spiral staircase. You know, the bases form the stairs of the spiral staircase. So there's and so usually if I would be drawing this, I would just draw a line for the sugar phosphate backbone and draw the horizontals. And each one of those would be would be a nitrogenous basis, like this. So as I mentioned, it's double-stranded. 2 polynucleotide strands coil around each other, form a double helix, like a spiral staircase. Bases along one DNA, hydrogen bonded bases on the other strand. So it's actually hydrogen bonding that holds these 2 strands together in that spiral form. Now, the functional groups hanging off the base determine which pairs pace up. So here's the thing. This is called the base pair rules, also known as Chargaff's rules. It's a guy named Dr. Chargaff figured it out. That A binds with T and G binds with C. A and T bind together with 2 hydrogen bonds. G and C binds together with 3 hydrogen bonds. So there they are. There's the base pairs. So each one of those CGs, TAs, ATs, GCs, those are called base pairs. And in human beings anyway, we have 46 chromosomes and we have 3 billion base pairs in those 43 chromosomes. And the order of those bases, like I said, that's the information, right, in how to construct a particular protein, right? And remember, hydrogen bonds, and this figure is a little misleading here because it has just 1 hydrogen bond between the base pairs, but that's incorrect. Remember, there are 2 between A's and T's, 3 between G's and C's. And remember, hydrogen bonds are the weakest of all the bonds, but if you put enough of them together, they give real strength to things, and they really hold together. These big macromolecules, like proteins, are really held together by hydrogen bonds too. And DNA is held together by hydrogen bonds. And so makes it a pretty stable molecule, and it lasts quite a long, even outside the body, it lasts quite a while. That's why, you know, forensic scientists can go into a room and collect DNA from a crime scene, for example, and still use it to figure out who was there. Because it lasts a long time. If it's frozen, it can last thousands of years. Like in mastodons that were frozen in Siberia for like 23,000 years. They collected the DNA from the cells of this mastodon. But not 60 million years, like Jurassic Park. It doesn't last that long. That's science fiction. Many similarities between DNA and RNA. They're both polymers of nucleotides. Both are made of nucleotides containing a sugar, a phosphate, and a base. You'll see the difference is that the sugar is different and one of the bases is different. Here's the differences between DNA and RNA. As its name indicates, the sugar in RNA is ribose, sugar in DNA is deoxyribose. Very similar. Deoxyribose just has one fewer oxygen than ribose. Instead of the base thymine, RNA has a similar but distinct base called uracil, U. So RNA is usually found in single-stranded form. So there's no base pairing. But DNA is always a double helix. Here's the thing, and also, there's one other difference also. DNA is DNA, and there's one form of DNA. It could have a different sequence of bases on it, but it's basically the same molecule. RNA, there are several different types of RNA. So it says RNA is usually single-stranded. Sometimes it's double-stranded. In some forms of RNA, it's double-stranded. If it is, then A doesn't bind with T anymore. A binds with U. So it's AU. So there's a ribo RNA nucleotide with ribose and uracil, different base. All the phosphate groups are exactly the same. Okay, the process of science. Does lactose intolerance have a genetic basis? So most lactose intolerant people have a normal version of the lactase gene. As I mentioned, lactose intolerant doesn't mean you don't have the lactase gene or you have a defective lactase gene. It just means that the lactase gene is turned off in a lactose intolerant person. So what is the genetic basis for lactose intolerance? The hypothesis is lactose intolerance can be correlated with a single nucleotide at a particular site within one chromosome. So later on in our course, when we get into more genetics, we'll talk about mutations. And mutations are error in DNA replication. So, when you're replicating DNA, the two strands are taken apart. And then a molecule called DNA polymerase comes along and reads these strands. And it reads the nucleotide. If it's a T here, it puts an A. If it's a G here, it puts a C. If it's a C here, it puts a G, and so on and so forth. And it's very accurate, highly accurate, but it's not perfect. I mean, nothing's really perfect. It makes mistakes occasionally. Usually only makes one mistake within the copying every billion base pairs. So, but that still means that on the average, every time you copy your DNA, you make a few mistakes. Usually those mistakes don't mean anything, but they can lead to different things. They could destroy a gene make a defective gene that codes, that has an error. It puts an amino acid in the wrong place in a protein, changes the shape of the protein, and it no longer works. That's how a lot of genetic diseases, we're all tested for genetic diseases minutes after we're born, like one called PKU, which is a inability to break down a particular amino acid. And it's due to an error in the parents replicating some DNA, and that happened to get into a gamete, and a child was born with this defective gene. Fortunately, that genetic disease can be treated. Not all genetic diseases are, death sentences or can't do anything about them. Some can be actually treated, and that one can be. So that's why we're tested for it. You could put in an incorrect nucleotide. Like say, if you see an A here, you're supposed to put a T here. What if you put a C or a G? It's an error. Or you could insert. So later on, we'll get into how a little bit more about these mutations, how these errors, what kind of errors can be made, and the results of the errors. But here it is. One single nucleotide is wrong in a series of nucleotides within one gene and one chromosome. Okay. Prediction, this site would be near, but not within the lactase gene. Because if it was in the lactase gene, then it might actually cause a lactase to be defective. But we know that these people who have lactose intolerant have a working lactase gene because they were given milk early on when they're infants. They're given mother's milk and they digest it, no problem. So it's thought that the problem is in someplace close to the lactase gene, but not in the lactase gene. And that means the regulation of the lactase gene. So this is called regulation of gene expression. And it's a really complicated thing. I know next to nothing about it, but it's really complicated. And there are areas between genes. Yeah, I should mention, genes are on chromosomes in a linear array. There's a gene here, a gene here, a gene here, a gene here. But there are spaces in between the genes where there aren't any genes. And people often wonder, well, what's going on there? Sometimes it's just gobbledygook. It's just junk. Sometimes, though, it's regulatory sequences. And that's, there's a lot of research trying to figure out how this works. If we can learn how we turn genes on and off, we could cure all kinds of human problems. And eventually, they'll probably figure it out. But like I said, it's really complicated. So there's a regulatory site that somehow these series of nucleotides hold the information to turn off the lactase gene after a couple of years of life. When you're finished weaning, it gets turned off and you're lactose intolerant for the rest of your life. So that's a regulatory site. So they examined the genes of 196 lactose intolerant people from 9 Finnish families. Research showed a 100% correlation between lactose intolerance and a nucleotide at a site approximately 14,000 nucleotides away from the lactase gene. So this had to be a site that turned off the lactase gene. And somehow an error was put in there that made that regulatory site not work anymore. And therefore it couldn't turn off the lactase gene. And the lactase gene turned on. And so even after you're an adult, you can still digest lactose. So there it is, human cell, chromosome 2, there's the lactase gene. And there's this section of chromosome 2, 14,000 nucleotides away, C or T. And I guess if it was C, it could, I'm not sure what that means, but I assume it means that if it was a C, then the lactase gene would be turned off as normally it would be. But instead T is there, which means that there's going to be an error in the message, and somehow now it doesn't turn it off. So most people are lactose intolerant no, they're not. Some people lactose intolerant. It's 80% of African Americans and Native Americans, 90% of Asian Americans, but only 10% of Americans of Northern European descent. So somehow this error was happened in our ancestors when they moved out of Africa and they moved into Europe. That's when this error occurred. So in cultures where dairy products were a staple in a diet, natural selection would have favored anyone with a mutation that kept the lactase gene switched on beyond infanthood. So this would be kind of like a beneficial mutation. Later on, we'll get into what effects do mutations have. Most mutations are neutral. They don't have any effect. Sometimes they don't even change the gene that they're in or the protein that the gene codes for. We'll see how that works later on when we get into protein synthesis, the genetic code, and that stuff. Second most common is deleterious. Usually you throw a random change into a well-oiled machine, you're going to cause a problem. But every once in a while, an error occurs that actually benefits the organism. And this would, you could think that this would be a benefit. Later on, we'll talk about natural selection, how the good changes are selected for in the environment, and they stay with a species from generation to generation because they're helping. This would have a help because now, before you couldn't really drink milk or have dairy products as an adult because you get really upset. I mean, lactose intolerance doesn't kill you, but it makes you really uncomfortable. But now you got a new source of food as an adult that other adults can't use. So you can see how that could be helpful, particularly when food is scarce, you know. And so these people had a new food source, and maybe they could be a little healthier and have more babies than the others that couldn't use this food. And this error spread. Okay. So ethnic groups in East Africa that rely upon dairy products are also lactose intolerant, but due to 3 other genetic changes that keep the lactase gene permanently active, lactose tolerant. So they found other errors in other places that could also keep the lactase gene permanently active in East Africa and in North Africa. Okay, well, here's a cave painting. I think we're through here with the, that's the Lascaux cave, I think in France, which used to be open and you can visit it, but it's closed now because people's breath were ruining these ancient pictures. Anyway, I think that's it. Yeah, now it just goes back to a bunch of figures. So there it is, chapter 3, molecules of life. And that ends the chapters that are going to be on the exam 1. And then we're going to move on to cells and how cells work in next group of chapters. So let me end the show, stop screen share. So like I said, I'll put this up in YouTube. Like I said, it's going to be 106 Life Molecules 3. Okay, see you later. Once I stop.